The overall goal of this experiment is to model the interaction of glioblastoma cells with microglia and macrophages during invasion in an in vitro assay.
So this method can help answer key questions in the tumor microenvironment field, such as which compounds or treatments can interfere with cancer interaction with macrophages, a cell type of the microenvironment, during the progression to malignancy. One of the main advantages of this technique is that it's relatively easy to perform and can be completed in a reasonable time and seems to accurately model glioblastoma invasion in vivo.
Begin by differentiating the cell line of interest using 4-ball myristate acetate as follows. First, plate 2 to 3 times 10 to the 5th THP1 cells in 1.5 milliliters of medium in a 6-well culture plate. Immediately after plating, add 100 nanomolar of PMA and incubate at 37 degrees Celsius and 5% CO2 for 48 hours. After 48 hours, remove the PMA by aspirating the media, wash once with 1x PBS, and add fresh media. Incubate for another 48 hours until cells are ready to use in the assay. Then, equilibrate matrix pre-coated chambers by placing them in a well containing 500 microliters of media without serum, and then adding 500 microliters of serum-free media to the top. Incubate the chambers for 2 hours at 37 degrees Celsius and 5% CO2. Next, gently aspirate the media from the culture vessel.